Okay, hello, my name is Kater. I want to talk with you again about neuroendocrinology and aging and about stress and more precise about glucocorticoid cascade hypothesis, which is written by Robert Sapolsky. You might know him. The plan of the lecture. Firstly, I will introduce you to the aging and how it is related with neuroendocrinology. Then we will make a short recap on hypothalamic pituitary axis, and then you will be ready to understand glucocorticoid cascade hypothesis and the paper of Robert Sapolsky that explores it. And at the end, I will just tell you where this hypothesis is used now and where it is used in the modern research, because the paper is quite old. So, first of all, a short historical reference. Neuroendocrinology, just endocrinology as a separate biomedical field, started in 1890s when Brown Stewart suggested that the animal tissue has so-called internal secretions, and he decided to administrate himself and some other guys with testicular extract of dogs and guinea pigs. And it could be a funny story, but it actually worked, and he started to feel himself better. And other people reported that they felt progress in cognition and everything. And the thing was in testosterone, and that's how endocrinology has begun. But only at 1960s, people decided to consider the role of CNS in all this regulation, because before the time, no one knew about the interrelation of hormonal and neural signals, some neurosecretory activity of hypothalamus, and only when all these facts were discovered, the neuroendocrinology and aging started to develop. So all theories of neuroendocrinology and aging is about the loss of homeostasis. You have endocrine system, some hormonal balance, and with time and with some events it is broken. We have some just observations about older people, and scientists try to somehow find the causation between all these facts and develop a new endocrine theory of aging. And yeah, when Jenga is falling, you die. There are five main theories of aging because of neuroendocrine system. First of all, we have to define aging, and here I will define it as the increase of death rates in population with age. We can actually discuss about the definition, but it is a linguistic trap, so we won't go here. The first hypothesis is hypothalamic dysregulation. It claims that all age-related changes in the hypothalamus are the cause of aging. The second is about neurotransmitter, and that there is an imbalance in the neurotransmitter in discrete brain areas, which cause aging, which cause death. And third is pituitary hypothesis. So the hypofunction, the bad functioning of pituitary accelerate aging and cause some pathology on the cellular level. Then we have progenia syndromes, and it is actually a separate field, and we won't discuss it at all. And the last one is stress theory of aging, which I want to talk with you about, and it claims that there are some intoxications in the organism, and there is involvement on your main stress axis, hypothalamic pituitary adrenal axis, and this altogether leads to the death and aging. So first of all, we have to understand what is the stress. There is a famous and classic experiment in neuroendocrine field when the dogs or rats are without adrenal or pituitary, they just remove these organs or have transgenic mutations, they could survive actually, but under stable stress-free conditions with some right support. And if you give them any stress, like exposure to cold or physical restraint or infection, they would die. Yeah, and that's because they don't have this regulation by the adrenal pituitary and proper response to stress. So when stress happens, you have three main stages. The first is alarm reaction, when you activate some defense mechanisms, then resistance, and then the exhaustions and recovery, when you collect all forces, all resources that you spent on the alarm stress. And this is the main axis that starts this stress alarm reaction. It's called hypothalamus pituitary adrenal axis. The first part is the hypothalamus that releases corticotropin-releasing hormone. Then it goes straight into the pituitary, which releases adrenocorticotropic hormone, and it goes straight to the adrenal that releases cortisol. And cortisol is a glucocorticoid hormone that's produced by adrenal glands, and it is actually used in just everyday life. In circadian rhythm, you need to feed to wake you up from your sleep, but it is also used for the stress defense. So when the stress happens, these axes start to work, and the cortisol that is released, any glucocorticoid that's released from your adrenal, they start some changes in the organism. They increase readily available energy and supportive metabolism, and decrease energetically costly anabolism until some less stressful time. So you have increased cardiovascular tone, altered cognition, the metabolism changes, but at the same time you are inhibited with growth and reproduction, and your immune system and inflammation is decreased too. But if you have too much stress, you have some bad disease like myopathy and steroid diabetes, infertility, and etc. So now I will talk you about, now that we recap all this stuff, we can talk about the glucocorticoid cascade hypothesis. It is actually a famous paper about it that I wrote. It's so Robert Sapolsky was not the first to propose this hypothesis. He just collected some experiments and checked two hypotheses. And actually, it is a really old paper. It's not 2002. It's like 1980s, and it's really famous. So it's like 3,000 citations. And at the bottom of the slide, you can see this scheme of glucocorticoid cascade hypothesis. If you have chronic stress, you have excess glucocorticoids. You have some which leads to generative changes in hippocampus, which leads to downregulation of glucocorticoid receptors, which goes into the feedback loop and increases the glucocorticoid production even more. And we will go through all these three steps, and I will explain the experiments that Sapolsky conducted and how they made these conclusions and supported them. First of all, we will talk about not about cortisol, but about corticosterone. It is common in rodents, and cortisol is for humans, more common in humans. They are almost the same, but they have one difference. Who will find the difference in the molecule? Oh, yeah, okay, that's right. Too fast. And corticosterone also plays a more significant role in some processes like electrolyte balance. So the first part is accept glucocorticoids. Sapolsky and his colleagues asked why do we have them? Did we have them? Yeah, this is actually a sad slide because this is the description of experiments they conducted. In his study, there were no some fancy transgenic experiments and all the stuff. They just tortured rats. And this is the slide about how they done it in different ways. And yeah, so there was a major acute stress. Mostly it was immobilization stress or cage transfer stress. The experiment was built in the way that they made rat being stressed. And then they measure the blood and measure the corticosterone in the blood after some time, with some period. And also they wanted to know if the chronic stress affects somehow the stress response and the glucocorticoid levels. So they also, some rats also received daily stressors, like 1 hour of immunization, cold exposure, injection of ether, or 1 hour of rotation and vibration stress. And they did it with rats for 3 weeks daily, and they also collected blood and measured corticosterones. I also wonder if the experimenters have depression because of this. So yeah, that's also how they measure blood with cardiac catheters. And what they found, first of all, is that and they conducted it with old and young rats. 3 months rat, which is young, which like us maybe, and 24-month rat, it's like old rat. It is almost here. And they found out that rats appropriately initiating corticosterone response. And by the way, I will call corticosterone B, just by B, because it's shorter. They also find out that old and young rats have normal circadian rhythms of B secretion, normal clearance rate of this steroid from their blood. And you might wonder, so what's the difference? And they found one very important difference is by how much the level of B is keep in the blood of the rat. So in young rats, B returns to normal rates within 60 minutes after the stress. But in old rats, it keeps its elevated concentrations for 24 hours after the stress. And here the top you can see is plot when they measure the level of corticosterone in the blood, and the dashed line is the old rat, and a solid line is young rat. So you can see how there is a difference in the period of recovery. Also, they checked does something change with the chronic stress. And when you give rats a chronic stress, they also have delays in adapting to mild stress. And this rise progressively. The more stress you give to the rat, the worse it reacts to the stressors, and the higher the concentrations are kept after the stress during the recovery. So that's how they made a conclusion that there is a substantial increase in adrenocortical output for the old rats. Then they also I found research that it is exactly the same for humans. So please don't stress. The problem next they asked Robert Sapolsky and colleagues why is it so that the old rats have elevated secretion of corticosterone and blood after the stress. They found out that in young rats, elevated concentrations of circulating glucocorticoids normally inhibit the stress-induced and normal-induced concentrations of glucocorticoid. So you have a loop where when you release some corticosterone and it inhibits the production of new corticosterone. And it is done majorly by inhibition of production of ACTH. But they found that it is not so in old rats, and they made the conclusion that rats have hypersecretion because of progressive loss of sensitivity of this HPA axis to negative feedback regulation. So Robert and colleagues asked why is it so? Why do you lose your receptors or sensitivity to all these glucocorticoids? And we have to go actually to hippocampus, which is not so expected. First of all, hippocampus and other organs have two types of receptors, mineralocorticoid receptors and glucocorticoid receptors. They are activated with corticosteroids, and the first type MRs are mostly concentrated in limbic structures, specifically in the hippocampus. Here's a scheme how these corticosteroids go through the membrane and activate these complexes, or change the work of the cell via the second messenger system. And what they found in their research is that hippocampus, which is involved in memory and spatial recognition, it has the high sensitivity, has the high density of glucocorticoid receptors, and it is actually respond and very sensitive to effects of corticosteroids. Also, they found out that each hippocampus sustains a loss of 50% of glucocorticoid binding sites, and at the same time, the other parts of the brain, like pituitary, hypothalamus, and midbrain, do not lose so much glucocorticoid receptors, and that's raises some questions. So they find out all these facts, and they checked all next experiments were conducted with so-called quantitative audio radiography with 3 DEX. DEX is a synthetic glucocorticoid. It's called dexamethasone, and 3H is a radioactive part that just radiates protons, and you can use long you can use camera to just catch these protons and make audiography of some parts of the brain. So they took the mouse, they cut the head off, and they took hippocamp and make a measure in the audiography machine. It's here at the top. Then they done some post-processing procedures and just count the number of receptors, MRs and JARs that were labeled in vitro by this 3H DEX molecule. Yeah. And here's a boring tables. No any plots here. They found out that in young rats, B here in the tables, it is actually important to us. It is the maximum binding of receptors to the 3D DEX. And they found out that in hippocampus in young rats, the binding was almost 2 times bigger than in old rats. They also checked it in other parts of the brain, and you can see that how hypothalamus have even bigger binding capacity when you're old. The midbrain doesn't change, and hippocampus lose the half of the capacity. They also, by the way, measure it exactly in the cytosol receptor levels. So they measure exactly the number of these receptors. And they find out that the receptor indeed are lost in hippocampus because of the loss of cytosolic receptor affinity to binding. So the capacity of the glucocorticoid receptor to bind to cell nucleus doesn't change with age, but the capacity of the receptor to bind with the corticosterone is changed. And they also asked whether it might be a glia, because glia have glucocorticoid receptors too. They again cut the heads of rats and made a special experiment, and they label only glia, and they find out that there is actually no age-related decrease occurs in glia binding capacity to the glucocorticoids, but it actually rises a little bit, which is, by the way, one of the biomarker of aging is glia hypertrophy. Yeah, and it is Barclaw's DEX is glia, and cortex is just the hippocampus neurons. So you can see how young rats has high binding. In glia, there is no difference. So then they wanted to check what exactly is wrong with hippocampus, and is this loss of receptors because of loss of the neurons. So maybe neurons are dying, and that's what happened. They again used quantitative autoradiography, and they found out, first of all, that the loss of neurons in hippocampus is anatomically specific. So not all parts of hippocampus are dying the same. And it's the pyramidal cells of layer 3A that is dying, and the other parts lose neurons, but not so much. And they found out that receptor loss is indeed due to death of neurons, but actually the surviving neurons having a smaller complement of receptors too. So it's not only degradation of the neurons itself, but degradation of receptors. And again, here we are interested only in this red boxed plots. You can see how the number of the bonding capacity is reduced in old mice, and this black mice is some transgenic mice that has lack in Z-protein and has the same symptoms as just old rats. Yeah, and for example, you can see that in CA4, CA1, there is no so severe change. And here's the example of this autoradiography. The left one is the hippocampus of young rat, and the right is the hippocampus of old rat. And this black dot is these receptors that are binded with 3H tags. And you can just by look see that there is much less black dots here. Yeah. So for now, let's make a short summary. And the Sapolsky and his colleague found out the corticosteroid hypersecretion. They also found out that It's because of the receptor loss in hippocampus. And after that, they asked whether this hypersecretion is somehow related to the receptor loss, or maybe there even a causation between these two events. And they asked maybe corticosterone hypersecretion just kill hippocampus and the cells. Um, and this is a scary slide with a lot of text, but the central part is important. They made a hypothesis, first of all, that hippocampus is a potentially inhibitory, has potential inhibitory influence upon the adrenocortical axis, and with the death of neurons in hippocampus, you inhibit the inhibition, and the corticosteroids started to release more and more. And they conducted experiments again in blood plots, and they checked whether the hypersecretion of corticosterone somehow affects the receptors. So they removed chemically the corticosterone receptors in hippocampus and checked what happened with the corticosterone levels in the blood of rats after stress. So they stress rats again. They find out that receptor depleted rats hypersecrete corticosterone after the end of stress, and when they normalize the receptor deficiency, the B secretion normalizes too, and if they reduce the medication with the normalization of receptors, the hypersecretion raises again, and it is all linear and parallel processes. So they asked maybe this is the neurotoxicity of corticosterone that might kill all neurons and receptors. And they found out that more prominent exposure to corticosterone indeed produced a permanent damage to the hippocampus. They took three types of rats. They have control, which were just sitting in cage doing nothing, and they took rats and some rats and made just acute stress with them, and they take third type of rats and made a chronic stress to them, and they found out after some time that the chronic stress rats has much less binding capacity, and even when you remove the stressors and give them time to recover, the binding capacity didn't come again to the normal levels. Here you can see it on the plot. The dashed line is chronic mice, and after some months of recovery, they have the same low binding capacity. But the control and acute stress rats has the high levels, and acute stress rats goes to the normal levels after some time of recovery. After some experiments, they find out that glucocorticoids actually not kill neurons in hippocampus, but it impairs the ability of hippocampal neurons to survive the insults, but not the insults themselves. So they took rats. They again make just normal control rats and stressed rats, and they gave them canic acid, which kills neurons. And they found out that rats with chronic stress has better ability to sustain this acid, and more neurons are dead after the vaccine. Here you can see in this table where controls are normal rats, experimental has higher use of receptors. And here, a short summary of all these experiments and what they found out during all these experiments. The cumulative exposure to high concentration of corticosterone leads to degenerative losses of neurons and receptors in hippocampus, and it is, in result, increase the corticosteroid concentration, and this is a feedback loop that accelerates itself. So when you have mild levels of stress, you have some motivation, arousal, and this is okay. But if you have strong and chronic stress, it causes neurotoxicity and kills your hippocampus. And that's it. This is the original image of the hypothesis. So you have this feedback loop of downregulation and hypersecretion to receptor loss. You have the stress that affect this loop, and all this leads to some bad symptoms of this feedback loop, like myopathy, muscle atrophy, osteoporosis, etc. And now, and that's like all. They have a lot of questions unanswered because it's old research, and they only use really easy experiments, just stress mice or rats. But nowadays, this hypothesis, glucocorticoid cascade hypothesis, is used in the so-called balance hypothesis. Yeah, that's how scientists try to find the interplay between CNS and the immune system, and how this glucocorticoid cascade might affect the inflammation, and they have theories about how chronic stress affects immune cells in general. I won't talk to you about that, but just look at this very complex scheme of how HPA axis, which at the top affects some cytokines and other endocrine organs, and they all together again would create a feedback group to make inflammation even worse. Yeah. And also, this hypothesis is used nowadays to understand the causes of neurodegeneration, Alzheimer's disease, and also Parkinson's, and many others. The main idea is that this dysregulation of EPA axis and cortisol release causes inflammation and neuroinflammation, and that's all together may affect and worsen the progression of diseases of the brain. And now, when you will look at this picture, you will know what it exactly means. This chronic stressful events, and this all result in the changes in microglia and accumulating of tau, and it all leads to some neurotoxic environment which can kill neurons in the hippocampus too. And that's all the links I've used, just for you to know. And this is the final meme. Thank you for your attention.